till... Det var i restaurangdatorer nu. Kan vi köra? Vi kan köra.
Hello? Is this microphone? Nej. Hallå? Hallå, hallå? Det hörs inte fortfarande.
Test, test. 1, 2, 3. Min funkar.
Funkar min? Funkar min mikrofon?
Yes.
Ja, jag har en fungerande mikrofon. I have a working microphone. A very, very warm welcome to this fourth DDLS conference. It's amazing to see so many of you here. It's really, really nice to have so many people interested in this interface between computer science and life science. And we have a full schedule ahead of us. And I would like to introduce to you our new DDLS co-director, Erik Christiansson. Erik is a professor at... So Erik is a professor at Chalmers University of Technology, and his research is focused on AI and computational methods for understanding infectious disease. We're very happy to have you here.
Yes, thank you. And I would like to introduce the other new co-director, Carolina Välby. So Carolina is a professor at Uppsala University, and your research interest is in image analysis and machine learning, especially with a focus on life science microscopy data. Very nice to have you here as well.
Thank you.
So Carolina and I, our task today and tomorrow is to moderate this meeting, and we will hopefully guide you safely through the program. So before we start, I just want to point out a few things regarding the program. So today we have two main sessions where we will have both long and short talks describing the different activities that is currently ongoing within the DDLS program. We will also have two poster sessions, one at 2 o'clock and one in the end at 4:30. And then during the first poster session, people with odd numbers will present their posters, while during the second poster session, people with an even number. So if you want to meet someone behind the poster, you need to check the number, and they should be present. I would also like you to try to respect the times during the program. It's, as Carolina said, packed, and this room is also packed. So please try to be seated when we start to avoid delays so that everyone gets their dedicated spot. With that, I would like to introduce the first speaker of today. This is Jan Ellenberg. He has a background as a group leader and faculty at EMBL, Heidelberg, Germany, but then he made a wise decision to move here to Sweden, and he's now a professor at Karolinska Institute. You have been the director for Scilab Lab for roughly a year, and since this summer you're also a director for DDLS. So please, Jan, the stage is yours.
Thank you very much. This is very loud, I think. But thanks a lot, Carolina and Erik. And I'm also very, very grateful that Carolina and Erik could be convinced to become co-directors of DDLS, because I think the program now enters an incredibly exciting phase of scientific output, production, networking, and really making new things happen. And I think it is impossible to run such a large program with 50 new investigators coming to Sweden in data-driven life science with one person only. And I'm really, really grateful they are joining the team. They now have the hardest task of being the co-directors in actually keeping me to time in talking about a program of so many people involved that has been built up at the national scale in only 25 minutes, which is a challenge. We could spend the whole 1.5 days on just reviewing DDLS build-up and successes. But before I do that, I just want to provide a little bit of context for where we are in life science and why DDLS is such a timely initiative. And I think in life science today, we really are at a transition in how life science as a discipline is working. And we are at the nexus of two convergent revolutions. On the one hand, we are in the middle of the technology revolution that really was at the birth of Scilab Lab in 2010 to realize all of the future of life science, will be driven by large-scale molecular analytical technologies in the future. We need to make those technologies available at scale to the community to take everyone along. And that's why Scilife Lab was founded. And convergently to that, these technologies came online, produced very, very large amounts of data, increasingly not only inventories, but with spatial and functional context information about the molecular underpinning of life. And we were drowning in this data. And from the beginning, Scilife Lab's efforts were matched in data production and data analytics. But we could never really scale this to the data production and to the mining of the knowledge inside it. But convergently in computer science now, we are also at a revolutionary point of that discipline, where tools from machine learning and AI have become scalable to deal with these very large and complex data sets. And so we are just starting to see the impact of that happening. And so that means in the future, life science is not just data-driven, it also stops being a descriptive discipline and it becomes a predictive discipline. We will actually be able to predict how living systems work, and by that also predict how they will function in the future and how they will adapt to changing conditions. And that's a revolution, I would say, for most life scientists that have been around like me for a long time, because we could never work like this. For all the young people in this community, this is amazing, because you can do life science in a completely different way. And that is really the context into which DDLS is coming in, as an extremely timely program to say we need to embrace this, and we need to understand the future of life science is data-driven, is predictive. What do we need to really empower the community in Sweden to do that? And so that's the overarching goals of the DDLS program that you are familiar with, is to develop the methods that are needed for that and to create this community so that the next generation of principal investigators, students, and postdocs can be recruited and put together in order to fulfill this vision, but also in order to take advantage of this unprecedented opportunity. So the overall objectives of the program are sketched out for a 12-year timeline in which we would like to have 50 research groups working in this domain, core funded by the program, but reaching out to the very large community of life science at large in Sweden, and also importantly to other scientific communities. So it's to first of all build the community by attracting the excellence in data-driven life science and train them, but also provide the data science and bioinformatics expertise and services that this community is going to need to operate in Sweden. And we just had a really productive discussion with the fellows about this morning on what concretely is needed to make this happen. And we are realizing that not everything is there yet, that you guys need to work. And we really need to start building these things very concretely now. But importantly, life science doesn't operate without computer science and data science. And so we need to link also these core communities with each other and create partnerships and impact so that also society benefits from what we are doing. And one way of doing that is also to engage in formulating policy and advise politicians with the results of what we do on what the action should be. So we expect impact on the science, of course, in a major way, increasingly also on applied areas such as healthcare, industry, and in general society, but also being able to put Sweden on the map globally in this area. And these things sound like high-level buzzwords that everyone dreams about that are not concrete. But we were just 2 weeks ago represented globally, at a meeting that the Chan Zuckerberg Initiative organized in California, and DDLS was presented. And this scale of an initiative coordinated towards data-driven life science is internationally also quite unique and is being noticed by our colleagues and partners in the US. So it's something really to build up and make work. Now, I won't go through all the highlights of what the program has achieved so far in its first 4 years on its 12-year timeline, but I think one of the key achievements is the community is now mostly there, not completely finished, but 36 fellows are here, and I think almost all of them are in the room. It's not that they are alone. They have already built a community in total of 175 people working on data-driven life science. And these people are doing science. They are publishing, they are making collaborations, they are formulating new projects, also together with the sister fellows communities in Scilife Lab and in WCMM, and with joint project initiatives with the computer science community in the strategic Wallenberg program was. So this is all happening. The research school has been launched. We have had students coming into the research school now, and the call is currently open to attract more students. And also the community is very active in organizing conferences. This is our annual meeting, but there are many, many other area-specific and topic-specific meetings and courses to bring people together. And we are also bringing the services online. And so I will review a bit these different areas in slightly more detail in the time I have today in terms of where we stand, but then also give an outlook. But this, in a nutshell, is what has happened in the first 4 years. So there are basically 3 major things that DDLS is trying to do. It's trying to do the research, build the community and empower it to do the research, help with education and training, and provide data management and data services for this community. And so maybe let's briefly look at these different areas where we stand and what we have achieved. So on the research side, the community is growing. It is established, I would say, more than 2/3. 36 fellows are here. You can see how they are distributed in the country and how this community is still going to grow to 50 with the extended financial volume of the DDLS that we have very, very gratefully received from the Wallenberg Foundation. And also now the program's very actively recruiting students and postdocs into the community that will, of course, also be in investigators beyond the core DDLS community to build a larger network. They operate in 4 different research areas, and I will come back to that. Now, the Fellows Program is quite unique in that the starter packages that these labs get for operating in Sweden are very generous. They are kind of ERC starting grant equivalent. So you have enough funding to build your initial team for 5 years. There's linkage to the hosting universities to provide tenure opportunities. And so one can really now engage in driving this paradigm shift towards data-driven life science at full speed at the beginning. And so that means there's a starting point for all of these colleagues that is quite unique for each of them. But I think also as a community, that really creates opportunities between these laboratories that are quite unique and that are only starting to appear and to be fostered. And so you can see in each area we still have recruitment opportunities, and the 3rd wave of recruitment in DDLS now will have a very explicit focus on AI in the life sciences and people who really develop and deploy AI tools. Because although this is a young program, only 4 years old, when it started and was conceived in 2020 and 2021, we were before the AlphaFold moment and we were before the ChatGPT moment. And it's sometimes hard to realize how quickly all of these things are happening. But clearly things are changing very, very fast in this field. Now, this community is very visible. We try also to make it very visible. And so if you're interested and you don't know them yet, here they are in short Q&A interviews on the web pages. So we try to introduce all of them. And it's very closely networked with the sister communities we have on the technology side and the SciLife Lab Fellows community for new technology and data production, and on the molecular medicine side with the WCMM program. And we have annual meetings again that bring all the fellows together, which was a fantastic meeting we had earlier in the year in the summer. So we have grouped the research in 4 different areas that very broadly represent life science and that allow to formulate common agendas and common issues and challenges in these different domains of evolution and biodiversity, epidemiology and biology of infection, precision medicine and diagnostics, and cell and molecular biology. And so I want to, because almost all the fellows are here, I thought I should bring a vignette of the research of every fellow, and then we really would be here for the next 1.5 days just in this overview. So I haven't done that, but I have brought 1 vignette from 1 research area, and I apologize to all the vignettes I'm not showing, because this is not a selection based on quality or on saying this is better than the others, but it's just to highlight the diversity of what's going on in data-driven life science in these laboratories. So if we look at cell and molecular biology, we look at the very core theory inside cells. How do membranes function? Can we model that? And can we use this knowledge to manipulate how cells function in the future? So really deep in the molecular mechanism at the cellular level. And if we, let's see if we can still move the slides. Aha, now we have data-rich slides and then the projector is getting stuck on them. So there we go. If we have data-driven epidemiology and biology of infection. One example again here is how does the microbiome composition determine outcome of pregnancy and what is the relationship between the bacteria in us and the core physiological function of the body. Again, a very relevant in physiological process, but operating now at a completely different scale of data-driven life science to understand how we interact with infectious agents in our bodies. We are going to get stuck on all of these data-rich slides. If we move on to precision medicine and diagnostics, then here one example, for example, is to look at pancreatic cancer and do this in an integrative way so that you really integrate data at the population level from translational and clinical research to understand much better how can you stratify patients, how can you reduce risk, how can you choose better treatments. So very tangible application and impact to healthcare and also to society. And finally, in evolution and biodiversity, a lot of it is about also creating the molecular understanding of the ecosystem. So it's really about measuring the biodiversity and especially longitudinally measuring this so we understand changes and then have the information available to model and to predict this even at the ecosystems level. And again, provide information for management of ecosystems, but also for policy. How do we protect the environment? So this covers the breadth of what's going on, and this is incredible. But it's also a challenge because these are very different areas of research, and sometimes the data and tools needs that they have can be quite different. So it makes sense to discuss in the different research areas, but it also makes sense to bring people together and to realize, actually, we all need genomics, we all need proteomics, we can do that together, but then we need different aspects of sensitive clinical data or of environmental data to bring together with that, and that's the challenge looking forward. Now, we have amazing firepower in DDLS, but we would always want to have more AI and machine learning and computer science competence. And so a major aim of the program also is to bridge the gap between the life science and the data science communities. And so luckily, there are two Wallenberg strategic programs that cover these two areas. There is the Wallenberg AI Autonomous System and Software program, extended also to the humanities and society, and DDLS. And so we have a strategic engagement with joint research projects, and especially an instrument for very large mutual research projects, the NEST projects. And so here we are now also in the phase where we are not just thinking about those collaborations or making calls for them, but we actually have the first 3 NEST projects between very prominent scientists from both sides to drive different areas of combining life science and computational science ahead. And we also will repeat this interaction and these calls so that we can really drive this together. But as importantly as interacting with our colleagues in computer science, I think is also to start to network our own community of DDLS investigators. And so for this, we started the Research Data Community Program to say what are needs between you that are shared and that you need to engage in jointly. Can you give us ideas for this? And we had a call for that as well. And again, we are now starting these projects. So here are the first 4 projects from the RDCP calls. And this is again something we want to continue to stimulate addressing joint technical challenges and joint data challenges between laboratories in this community. So I want to briefly say also something about education and training, because scaling this revolution in life science towards data-driven requires to train lots of people in new methods and new computational knowledge. And so how do we do that? And so for this purpose, the DDLS Research School was launched in 2024. The idea here is to select the best data-driven projects and PIs across Sweden, so this is not limited to the DDLS fellows community, and then recruit nationally the best students from the global pool and really provide education and training activities to prepare them for a career in data-driven life science. And so this is starting now. With the first students are there. The next call is open. And I think a key here, again, will be to build this community so that it doesn't fragment and get isolated in individual places and laboratories, but that it really bring people together by joint networking and training activities so they can collaborate and solve problems as a community rather than in isolation. And so the research school now will also contain the postdoc program. So Lukas Fjell is the lead of that, and we are about to launch it. We had a great discussion yesterday in the DDLS steering group on the model for the postdoc program, which will be a 2+1 year model for fellowships. And again, this will now be coming online, go through the SciLife board, and we will have calls also for the postdoc program with a very similar vision as the PhD program to bring the best computational people at the postdoc level into Sweden, into the most exciting projects and laboratories. So a little bit is worth also saying about data management and services, because all this individual research activity needs to be underpinned by data tools, data infrastructure, and data availability. And I know this is not where it should be, especially in Sweden if we talk about medical and health data. And so we really need to do something about that. At the same time, we do have a strong infrastructure that we can build this on in SciLife Lab, where we have both the SciLife Lab data center that engages in IT systems and infrastructure, data sharing, open science, data management, and hosting the models and the computational tools that people need to run. And on the other hand, we also have the bioinformatics platform that helps people in all areas of technology with developing the data analytics that they need, because you can't be an expert in all data types. Typically, you dive deep into one particular area of machine learning and computational science. And so both of these units have dedicated staff for DDLS and for the different research areas, but they are both part of a bigger system of data science infrastructure that SciLife Lab has built up that now totals 176 people across the country that provide data services to the life science community in Sweden. And so that's a very strong foundation. I know it's not delivering everything you want, but there are lots of very good people that are there to support, and it's really also our role to formulate the challenges for what are the future services we need, or to develop the tools that can become the future services for the community at large. And so that's an extremely strong basis. Now, SciLife Lab's data output, I think, is another reason why DDLS is placed so strategically at SciLife Lab, because combined, all the different technologies at the SciLife Lab infrastructures produce 20 petabytes of quantitative molecular data a year. That's a treasure of data for mining and knowledge extraction. But it often is a bit siloed in the different technology areas, and that's something we are really changing now in SciLife Lab, where we are integrating the data services between the data center and the bioinformatics platform so that data from any modality is interoperable with each other. So this verification of data that everyone talks about, we're actually now putting into practice, and increasingly we will need to use AI to be able to do that, to be able to integrate and then mine the data. And so Ola Spjut has spent the last year on developing an AI strategy for SciLife Lab and DDLS to say what are the most important areas where we can deploy AI and make use of AI. And he has formulated 4 pillars to look at our data ecosystem and make it AI ready, to make sure the compute infrastructure is available to run AI at scale, to be on the constant watch for new AI services and tools, because this field is moving extremely rapidly, and also to engage heavily in training, because we can only scale this by educating people to use this. So this combined is now leading to a big push in better data services across SciLife Lab, and in a large way also to support this community, where we are hiring 15 new data scientists into our data platforms that produce data, and 5 new AI engineers into the data service platforms. So we're coming to the end, and I see Eric becoming concerned that his co-director role is already becoming very tough to keep me on time. But I want to give a little bit of an outlook where we are going, because we could say this is all amazing and so successful. Let people just do the work that they need to do in the individual laboratories, and we are actually done in establishing the program. But I would say the fun is only starting. If you look strategically at what DDLS was supposed to do over the 12 years, it is in a major way a recruitment program at the level of the fellows and at the level of the postdocs and the students. But of course, the research activities, the data service activities, and the impact will carry on through the 12 years. And so we are really at a turning point now after 4 years where the recruitment phase is still ongoing, especially at the junior level. The research schools are still in the build-up phase, and the fellows recruitment now will be completed by 27. But we're really transitioning to the output and the impact phase. And what kind of output and impact we will have will in a major way depend on how we organize this community and how well we empower it with support and with networking activities. And so that's only starting now, because only now are the investigators there. They have defined what they want to work on, what the key problems are, and what tools they are going to develop and need. And so this is now the scientific opportunity. I would say here was a lot of setting it up and getting people in, and I can think we can be amazingly proud of that achievement and also the quality of people that we have gotten to Sweden. But now it's about doing the science and doing a lot of it also together as a community and not only individually as individual students. So that's incredible, and I'm really, really looking forward to the years to come, because we will have a lot of opportunity. And so we are in the third phase of the program. Wallenberg has organized the 12-year funding into 5 phases, and for every phase we also need to feedback to the funder and the foundation, what do we want to do in the next phase? What is the major objectives? And so I think an overarching goal is really to attain leadership in life science AI. This is a pretty bold statement. But I think AI in general is, of course, something where Europe at the moment, rightfully, I would say, is behind, because we don't have the data center capacities that our friends in the US and in China have to be leading in large language models. But if you think about AI in life science, we are not necessarily talking about large language models, because living systems that I'm trying to interact with molecular don't speak English to me. I can listen a long time, but the cells don't tell me in English how they are actually working and why they are doing something. We need different models in life science. We need models that explain something and that can deal with the physical and chemical reality that life operates in and with the molecular language that they speak. And these models don't exist by and large. We have to create them. And this is an opportunity where leadership is still possible with a concerted effort. And I think we really are in a unique position with this community to go there and go for that. So I think that's a very high-level goal. Now, how to get there, of course, needs concrete measures. And so, as you saw, we are now launching the postdoc program. These will be key, more experienced people in AI and life science that will drive the projects in the laboratories. The research school will grow. We are in the annual calls now for the PhD schools, and we will also have regular calls for the postdoc school. That means we grow the community and we strengthen collaboration. So we also want to really reinforce the instruments we have now for multi-PI activities in the program and for linking the PIs to the infrastructures on the data side and also on the experimental side. And your input into how we do that is extremely welcome, because we can shape how we empower this community in the future. So that really should promote the excellence across the community, that we really aim high. We aim for the best science and for the most impactful science, because we have the tools, the technology, and the intellectual capability to do that. And then really by that also strengthen Sweden's leading position in life science data services, because the new tools we create now will become the services of the future for the community at large, also internationally. So that means this excellence of research and service will be translated into sustained impact, because that means we are getting into fortuitous positive feedback loop of new research, creating new tools and services, maintaining those and sharing them at large to the community, which creates a lot of impact, but it also, especially in the areas of medicine environment, will lead to a lot of translation opportunities for more applied areas and a lot of technology innovation, new companies, new services. So these are the high-level goals. It's a long list of bullets, but that's quite an ambitious description for what we want to do in the future in DDLS. And then the question is, of course, are we set up to do this right? And so this infographics tries to encapsulate how this can happen. Do we actually achieve this vision? So this network, you can imagine the DDLS fellows to be, that they are an interlinked community. Now Eric is getting up to shut me up. But how do we empower them beyond what the individual laboratories do? We have the RDCP community projects, we have the NEST projects together with computer scientists, and also we have the technology development program with SciLife Labs, so that all the SciLife Lab platforms, data or technical services are available for collaboration. This is extra funding that goes into these collaborative, community-oriented projects. It relies on the data service infrastructure, but then it really creates in all of these areas a lot of impact. And so I think for the future, the challenge is to enable DDLS to be more than the sum of its part for creating impact, to transition now from recruitment to output and science, and really empower this community as we can, and think also about new strategic collaborative instruments. I'm really happy here today and tomorrow to talk about that and listen to your ideas. So in closing, I just want to acknowledge that none of this could happen without amazing people in the organization and the steering. You already heard the new co-directors introduce each other. I don't have to do that anymore. This is the DDLS steering group, very dedicated colleagues that try to guide the program, but now increasingly the program needs input from its community that now exists. It's no longer being built, it's actually here. And we talked this morning about how we are going to do that. And if I can advance to the last slide, we also have adjunct members to the steering group. And the most important slide to show is this. We have an amazing support team in the DDLS office and also in our data center and bioinformatics platforms that support the program, and you all know them. Without them, nothing would happen, and these people deserve the applause that you might give now. So DDLS is incredible, the future is bright, but the opportunities to realize them require all of us to really work together and to shape this future together. Thank you.
So thank you, Jan, for opening this conference. If you have questions or want to discuss topics with Jan, he will of course be available during coffee breaks, so don't hesitate to approach him with your questions.
Yes, so next I would like to introduce the NEST projects. So NEST stands for Novelty, Excellence, Synergy and Teams. So these are 3 5-year projects that got 30 million crowns each. And they all address specific strategic high priority research challenges. And that's where the novelty and excellence comes in. The word strategy points to the fact that these are projects are a collaboration between DDLS and the Wallenberg AI Autonomous Systems and Softwares program. So we are happy here today to have the teams, or the team leaders, as the T in the NEST, that we have one team leader for each of the projects. We have a team leader from DDLS and also a team leader from the WASP side. And because of the tight schedule, I will actually let you introduce yourselves. So please, Rosio and Ola on stage.
I don't think you hear me. Now you hear me. Very good in that sense. So 5 minutes is fast. My name is Ola Spjut. And her microphone is not working.
Now my microphone is working. I'm Rosio Mercado Oropesa.
Yes, and we are the PIs of Timed. So with Timed, we asked ourselves the question, how do cells evolve over time under perturbations? And there's a lot of important processes that requires looking at cells over time in that principle. So this is currently understudied. A lot of the omics that we use currently is destructive. There's a general lack of temporal data sets, and because of that, also there's a lot of AI methods that needs to be developed to work on these projects. So in the TIME project, we'll study the dynamics of cancer cells using live cells and multi-channel microscopy. So here you see the team so far. We are 5 PIs, myself, Rosio, Ashkan, Prashant, and Brinton. And we've already recruited 3 PhD students. I think they are all here today, and there's ongoing recruitments in that sense. So we can dim the light. I think that makes it look a little bit better. Yes, exactly. So anyone who's worked with cells knows that sort of, and that most of the studies we do, we look at cells at the very specific time points, and there's so much that we cannot information that we cannot capture from this. So. But one of the challenges is also to scale this up, and this is some shots from my lab where we worked on how to scale this up using automation, and that could also mean that you put microscopes inside the incubator, or you put the sort of stage top on the microscope. So building this technology, we've done this for quite some years, which enables us to sort of scale this up. And I think in contrast to many other consortia where they come together and make a huge, large effort and produce a data set that's then analyzed during several years, we're going to continuously work with producing new data iteratively in this project. And with that, I hand over to Lucio.
Great. So I just want to go over the objectives here for the project. So one of the goals of the project is that at the end of the 5 years, we would have created one of the largest temporal multi-channel imaging data sets of perturbed cells at the single cell level. Then we would also be building uncertainty-aware temporal foundation models and a sequential design framework for efficient experimentation. We would develop generative AI to help us propose molecules or combinations of molecules that would drive desired temporal phenotypes. And we would validate these methods and interventions in patient-derived ovarian cancer models while releasing data, code, and protocols openly. So here again is those of us who are directly involved in the project. At the moment, we have a poster number 4, which you can come to during the poster session if you would like to learn more or get involved. One of the things that we've discussed actively since the start of the project is that we want to be very open in this nest. So if you're interested in collaborating, we would love to hear from you. So with that.
Yeah, and I think just underlining that, if you're interested in sort of working with the data that we produce, then please reach out to us. If you have some cell models, are interested in live cell, well, come and talk to us. I mean, we cannot promise you resources, but we promise that we'll really like to work with you, within the context of this project. So, thank you so much.
Thank you very much. And then next on stage is Thomas and Johan, please.
Thank you, Carolina. So I'm Johan, and the DDLS PI here. And we have a project about learning 3D genome dynamics from heterogeneous data, starting with bacteria, so kind of toy model. Eventually, the technology can be used also for eukaryotic cells. So we want to figure out the rules for the structural dynamics of the bacterial chromosome. And we want to do this to learn how chromosome dynamics and the spatial positions of genes impact gene regulation. And to do this, we combine 3 very high-dimensional types of data that give you different information. So, for instance, we have developed the method to monitor, so we put in labels in the chromosome of cells and track them over time, and we can do this in thousands of different locations at the same time, and then read out which one is which by in situ genotyping. So this gives you kind of distance constraints for the dynamic structure of the chromosome, but you only get it for one distance at a time. So then we combine this with other techniques that gives you, for instance, if you fix the cells, you don't get any dynamics, but you can then use Hi-C. It's a method to see which DNA pieces are near each other in 3D space. And you get all these connections, and we can do that for one cell at a time to get a kind of dense view of what is close. And then finally we can look at individual cells, and with a few hundred markers maybe look in those particular cells where these sites are at very high resolution. And then the challenge is, of course, to combine these types of data with different sources of errors and different types of information into a common model. And here I hand over to Thomas.
All right, so I'm Thomas. We should also mention we have Magda Blenklo in the project, but she's in Italy as we speak. So my part in the WASP side of this really starts from this picture here. And as Johan said, we have different kind of data that represent either dynamics or no dynamics. Maybe a few, maybe many. So this is something that I bounced into in the automotive industry roughly 20 years ago, where you have, for example, a radar sensor that sees certain things and a camera, and they complement each other. So if you can merge that information, you get a much better output, which is what's now sitting in your cars. So very much the same thing happening here, but of course, very different setting, very different sensors. The mathematics is exactly the same. So we'll try and leverage a bit of that. And if you click to the next slide, so a few of the approaches that we will make use of is, for example, diffusion models, which underlies most of the generative AI that you know of today. And that model can be used not only to generate data, but also to understand data, for example, this type. And that is something, that's a thesis I have, or a hypothesis, I should say, that is severely underutilized. So we'll try and change that over the coming 5 years. I also outlined something called optimal transport, which is a mathematical problem that has several hundred years on its neck. But we had some very recent breakthroughs in terms of solving the computational problem, allowing us to use it. And why am I talking about that here? Well, because as Johan said and showed you in the movies, we have a situation where we have hundreds and thousands of cells, but they have something in common. And optimal transport allows us to reason about that in a probabilistic fashion. So we will use a little bit of that as well, but we'll start from what's called SLAM, so simultaneous localization and mapping, which is from robotics. And that provides a mathematical framework that fits the first kind of measurements that we have access to. So I think I'll stop there.
Thank you.
Yep, thank you.
Thank you very much. Very, very exciting. And the next pair on stage is Matthias and Claes. Welcome.
Thank you very much. So first of all, we're really happy to be here today to give you a brief overview of our NEST project, AID4BC, that focuses on precision cancer medicine in breast cancer, primarily. And this is an overview of the PIs and the teams that are part of this NEST. So we have myself, together with some other co-investigators at Karolinska Institute. I lead a group focusing on predictive medicine, working on AI, primarily in pathology, but also in other applications. The other DDLS PI is Sofia Zachrisson, who is a professor in radiology in Lund University, and also driven both clinical trials and other AI-related studies in that domain. And then on the WASP side, we have you, Claes.
Yes, on the WASP side, we have Linköping University with myself and my colleague Daniel Jönsson representing the visualization and human-computer interaction side of computer science. And then Uppsala University with David Zakaria and Jens Sjölund with expertise in topics such as modeling causality and uncertainty. And to complete the picture, we also have collaboration with industrial partners Sectra and Stratipath in this NEST.
So the main idea that we're addressing, or the main problem domain, is in cancer precision medicine. So the challenge is to identify patients for specific targeted therapies, but also to identify patients with, for example, worse prognostic outcomes. And the unique kind of approach we take in this NEST is to work with multimodal data. So we have data of different types that come from the healthcare domain. So radiology, pathology. We also have RNA sequencing, molecular profiling data, and other clinical data from national health registries in Sweden. So it's part of a kind of longitudinal also trajectory of the diagnostic pipeline for patients, going all the way from screening towards diagnosis and various kind of treatment decisions along the way. So what we're doing here is building enormous data resources, I'll come back to that later, of very large size, multimodal, and then we're developing both applied models for patient stratifications addressing multiple decision problems in the area of breast cancer, and we're also developing foundational methodology for this, and also addressing the kind of clinical decision problem. So the aims are essentially these targeted kind of applied methods development to predict outcome for the patients, and the opportunity we see is that AI can also allow development of scalable solutions. molecular diagnostics is very expensive. AI solutions based on some other routine modalities could also scale this in a different way, also in other parts of the world, which is attractive. We're doing focused methodology development in the AI domain in several areas. And then, of course, we're also having to address the issue of data models and infrastructure to facilitate both this product, but also the translations towards healthcare domain. And an important part, of course, that we need to address these kind of questions and do this method development in the context of very large data sets. So the field, precision medicine, is often constrained too much by the number of observations. And here we are looking at, we will have 10,000 patients with multiple modalities in this project available to us, which open up completely new opportunities. And what we can say here is that it also builds, this data sets build upon existing resources in these different partner sites that we bring together and also expand during the course of the project. So we have a starting point that we're not starting collecting data now, we have a lot of the data already available to us. And in respect to the kind of opportunities for the nest here is really the synergy effects that we can have between the DDLS and the WASP sides. And the DDLS partners here are quite technical already. They're doing a lot of AI development already, but there's definitely a supplemental kind of concept there. So the applied side, more DDLS responsibility, and then we have the methodology side.
Yeah, so on the WASP side, we are, as you see, partly responsible leading the work on data models and infrastructure, and among other resources working with the ADA data science platform, that part of SciLife Lab that you heard Jan mention before. And also the computer science methodology development, really starting from the actual problems in the clinic for future clinical decision support. There are true fundamental challenges that we need to solve in terms of understanding how to work with uncertainty and elicit causality, and also interaction design in the future world of agentic AI.
And the impact we hope to have is in several areas, quite obvious, I guess, both scientific impact, of course, that we can get conclusive insights from the work we're doing based on the large data sets and the kind of methodology approaches we do. And we hope that this also will be a starting point for translation and the kind of work towards clinical implementations as well. And of course, we hope to contribute to building this, community as well that we heard Jan and others talked about in Sweden through this project. So thank you very much.
Thank you.
Thank you so much. I think, is it not? Yes. Thank you so much. I think these were fantastic teasers, and I'm very much looking forward to hearing what comes out of these projects. And it's also nice to see that you're inviting others to take part of the data and contribute to the developments. So now I would like to introduce our first longer scientific talk this afternoon. So please welcome on stage Kelly Swartz. Kelly is a DDLS fellow at the Swedish University of Agricultural Sciences in Umeå. And you will tell us about climate adaptation in natural forest trees. Please.
Yeah. So thanks very much for the invitation, and I'm really happy to talk about the work that my group has been doing over the last number of years. So I don't think I have to convince, hopefully, anybody in this room, but climate is changing. But the real problem is not so much that it's changing, but that it's changing very rapidly. So it's changed as much in the past 100 years as it has over the past 10,000. And that's problematic if you're something like a tree in the forest, because if maybe you live for hundreds of years, you were selected under and adapted to a very different environment than you're currently living. This is a problem. Trees not only have ecological, economic, and cultural value, but they're also a major carbon sink. So if we want to sort of stop the feedback loop, we should probably keep the trees in the forest alive and photosynthesizing. Conifers, which I work on, have a very long history. So they radiated over 300 million years ago, and this was a very different Earth than we're currently experiencing. So it was much hotter than it currently is, but not actually much hotter than the future climate projections or some of the worst climate projections. And so they probably have the adaptive variation somewhere within the genome. They've lived through these hotter temperatures, and it's probably retained. They have very large effective population sizes, which maintain high genetic diversity. The problem is that this diversity is not evenly distributed. So this is a common garden experiment, so everything grown in the same location in British Columbia by Sally Aiken, and all of these trees were provenance from up and down the west coast of the Americas. And you can see that even though they had the exact same environment, the ones from the south grew much faster than the ones from the north. And so the problem here is that all this adaptive variation is then nested within population structure, or they're confounded. And so if we want to understand or identify the most adapted individuals for future environments, maybe when we look at a tree, you can see, sort of how it grows or measure a number of traits on it. But the problem is that this could be either due to the genetics underlying the growth, or it could be due to the environment that it's experienced. And so maybe a better question to ask is, can we isolate genetic from environmental effects on growth? The problem is that trees are really not a great model system. So we can look in the natural forest. The problem is that we can see that some grow more than others, but these are also in unique environments, and they've experienced each unique different environments. This is not unlike humans, actually, incidentally. And so the problem with these systems, what we do in crops, is basically use replication as the key to isolating this heritable variation. So we can replicate individuals, and with proper randomization across enough environments, you can start to parse this variation into a genotypic component. So that's common across different environments, an environmental component, that which is purely environmental, and the interaction between them, or the plasticity, G by E. And you can do this in trees like you can in crops. It's effective, but it is slow, and it's limited to the number of environments and genotypes that you can evaluate. Luckily, we have this cool physiological aspect of trees where they record their annual growth as tree rings. So basically, they grow a certain amount starting in the spring in the temperate regions, and then towards the end of the year, they sort of stop growing, and the cells thicken, and they get smaller, and you can sort of see this as this brown line in an increment core. They've lived through a lot. So we can look over centuries that the trees have been alive. You can see basically the 16th century megadrought produces much smaller rings than the tree was growing typically in its life. So another way to think about tree rings is what it gives us is longitudinal measurements that replicate the genotypic responses across the years, and the years here being the environments broadly. And then we can further parse this into the biotic or abiotic components from, say, weather station or satellite data, as well as more historical or biotic events that you can measure from past historical records. And so we've started working on this in Norway spruce, although you could actually do this in any temperate tree. And we do this mostly because it's everywhere. So it has this really massive both native range and also managed range that's centuries long. And so the way that we've been sampling is designed to really capture every level of environmental variation that can be experienced. So we sample broadly across Europe, and actually this only goes till 2023, but we've also started sampling now in Scandinavia. This captures climatic, sort of broad level Mediterranean versus Alps. Within an area, we sample across the regional environment, so high altitude, low altitude, north slope, south slope, et cetera. And then what I mean by sort of these plots is that we are actually sampling we're sampling up to 10 centimeter accuracy every sort of tree within a 26 meter radius. So we really have fine grained mapping to capture this microsite variation or neighborhood effects. When we sample, we take increment cores, we take tissue for DNA, and then we have a bunch of metadata on the trees. And this generates a lot of rings. So we've developed a lot of automation techniques to measure and sort of validate these millions of annual rings. We have sort of nice microscopes. We can use CNNs for ring detection. These are not totally perfect, so we also have Shiny apps that we've developed for both fixing any residual errors from the CNN, but also doing what's called cross-stating, or making sure the year is correct for the ring that is assigned. We want to do, in the end, association genomics, and so we've sort of built approaches for doing scalable libraries because we have a 20 gig genome, so this is also sort of problematic. We can't just do whole genome on these things. So Miguel and the group has developed a lot of these approaches. They work very well. We get about 2 to 5 million markers, generally covering the gene space. And they're also unbiased, so we can do population dynamics as well. So looking at this, if we're trying to isolate adaptive variation, we can do this in a mixed model regression framework where annual growth is the response. So that's how much a tree I grew in year J, et cetera, et cetera, et cetera. We can parse this into the tree term. So this is just identifying tree 1 versus tree 2 versus tree 3. This is sort of a growth or yield term. We can use, in this case, the environment is just set as which year were you growing. So it's the most expansive version of the environment. And then you can look at the interaction between these. There's a bunch of other things we need to take into account. So which core it came from, which species it is, because sometimes these are mixed stands. And then a bunch of sort of things that we need to deal with because these responses are not independent. This is not actually a totally kosher model in a lot of ways. So we have microsite variation. It's like if you had an agricultural field and you never re-randomized year after year after year. That's kind of our situation. So we can deal with this using Euclidean distance matrices or two-dimensional splines. We include autoregressives to deal with sort of buffering effects. So starch reserves, if you have a bad year after a good or a bad year before a good year, or after a good year, then you might have less of an effect than if you have a series of bad years. Additionally, photosynthetic capacity is retained for about 6 years in spruce. So actually, how much you grew 6 years earlier is still contributing to your active sort of sugar processing. We also have this kind of just geometric problem where when you put biomass on a column, if you have a small column, it makes a bigger ring than if you put it on a big column, so you have to account for this as well. Okay, so a lot of these terms are nested within a plot. So because we actually end up with a ranked efficient model, we actually process on a per plot basis before combining across. And so when we do this, looking at, say, plot 83, which is from Mercantour, France, near Nice in the Alps, we can see that our variance is sort of parsed out like this. So a lot of the variation is purely environmental. This is not heritable variation. A lot of it is actually also thrown into the autoregressives, basically control a lot of this, and that leaves us with about a third of the variance being heritable, that from the tree and the tree by year term. We can estimate some kind of loose broad sense heritability of about 0.3. This is nice, but the thing is that, I already said that adaptive variation is population specific. And so what we really want to do is compare trees across Europe. So it's nice that we can do this within, but can we compare across? So the way to do this, then, is to really focus on the heritable fraction of this variation. We don't care about what's purely due to the environment that you've experienced. And so we can isolate this heritable variance, or the tree term and the tree by year term, and basically make a new sort of term out of this. And we can bring this forward to combine across trees across Europe. And so this is just looking at 11 plots from 3 distinct European environments. They're genetically differentiated, but not all that much. If you can see the coordinates on this, it's only explaining about 4% of the variance. There's not a lot of differentiation in spruce. We can run these plot-specific models and isolate the heritable variance from each of these plots and move forward with this as our new response. So now we have this. Now we can look at the observed environment, so not just the year, but actually, sort of, for example, the maximum temperature in March. This is an interesting term because across all of these years, this is when the temperature reaches about 5 degrees, some years, but not others. And this is the physiological growth minimum for spruce. So it's a good way to sort of say this is the start of the growing season. We now have a plot term, so this basically removes variation that's associated with, top of the hill, bottom of the hill, that type of thing. We again can model the growth on a per tree basis, the interaction term, but now this is the growth conditional upon the temperature that it experienced in March. And I should just point out that this pilot data set is only 782 individuals, but this is about 47,000 observations. So there's a lot of data going into this, even though there's not that many individuals. So basically the G by E term on this is our phenotypic plasticity or adaptive variance. If you combine this with the tree term, this is basically adaptation to specific environments. Okay, so now when we look at this is just, again, the March temperature sort of flipped on its side. You can model the plot variation, kind of makes sense. But what is really important here is if we look at the growth conditional upon the March temperature, these are all centered. So basically, even though these were all estimated together, I should say, and then I sort of split them apart based on plot just to compare basically how comparable these estimates are. And we find that they're all sort of centered along 0. So this means that we get comparable estimates out of natural forest trees, and we can actually use these to combine across in a way that won't just be confounded. The growth term, I should say, so this is the tree term, and this still has, this is not centered. So there's still basically population structure in this. If we add genetic markers, so we haven't had any genetic markers in this model yet so far. Now we add a kinship matrix or a genetic relatedness matrix to estimate narrow sense heritability out of these estimates. And you can do this across many different environments, not just March temperature, and we get sort of a range, but they're generally quite high. If we look at March temperature, we get an estimate of 0.73. So it's really, we are isolating adaptive variation. If you do a GWA on this, so this is just a GLM, a QQ plot from a GLM, and you see that not only do we not have, too much population structure, but we also have real signal coming off of this. I should point out that this only works because we are using this heritable variation. So this is what I just showed you, where we have sort of this nice centered estimates across the different plots. But if we do a naive model where we don't remove the environmental variation, it looks something like this. So these are not centered, and this shows up in the association genomics. So in a QQ plot, if you're just using the heritable variation, it's the one I showed before. If you're not, basically you have a ton of population structure, and you also don't have any power to associate anything. This is just for fun, the Manhattan plot from the one that's good, sort of shows it's working. But in general, we can reduce population structure and increase power by parsing these models. So what I've shown is that we can extract comparable effect estimates with power for association genomics. We can do this from any forest tree genotype in a few minutes. We can do this with tens to hundreds of environmental observations. And we can do this, importantly, for discretized environmental responses. And the reason this is important is because as environment changes, it doesn't change in a sort of a steady state. This is just showing predictions for temperature and precipitation across Europe and in the future. And the important part is not the details, but the point that they're not the same. And so we need to be able to model all of these temperature, precipitation, et cetera, changes independently if we want to be able to predict which tree to plant where in the future. And just to sort of follow up, some preliminary prediction modeling shows that it's sort of generally working, but we still have some work to do. So this is just using a kinship or a genetic relationship model to predict the growth response. And so in cross-fold validation, this is 5-fold cross-validation across different locations with a couple different models that aren't important. Basically, we're doing quite well. So we're getting above 0.9 in cross-fold validation. If we try to do it and leave one out by country, it drops quite a lot. The good news is that we're doing better than the naive model. The bad news is that we're still not, we don't have that great of accuracy. But it is better than 0. So we can also do this using in a G by E predictive model. So here we have tree plus the tree by year term as the response. And then we're modeling it both with the kinship, and then also the kinship interacted with an environmental similarity matrix coming from, in this case, these are the RCP climate predictions for the future. And so we can make an environmental similarity matrix involving the environments that the trees experienced and the environments that we're trying to predict into for regions across Europe. We can take, for say, example, this is, I think, tree 38 in plot 31. And we can start to make these kinds of plots where we can say, okay, in 2100, this tree that's currently living in Berchtesgaden, Germany, will basically no longer be able to grow in Berchtesgaden, but it will have a better ability to grow if you move it a bit northeast, and actually, weirdly, maybe by Umeå. So, these are the kind of things we can do. I wouldn't put too much stock, per se, in these models, but this is where we're going. And a lot of people have gone into a lot of this work, and so I want to thank my really excellent group, everyone in Umeå, on both the UPSC side and also at SLU, and also DDLS and SciLife for all of your support. So I'm happy to take questions. I don't know if we have time left. I guess a couple minutes.
We have plenty of time for questions. Thank you so much for a beautiful talk and so exciting to see this. So do we have any questions from the audience? Please raise your hand and you will get a microphone here from us. So I would like to then start to ask you, so this is, I'm not at all into trees, and when you started your presentation, you just feel like, God, these are growing so slowly. How can you ever study them?
Yeah.
But then it's amazing to see how much you can still get out of it, and the historical landmark that you have in the rings. But how much of what you, like models and things that you have come up with here, can you bring to the other fields of life science, and how much models do you bring in to your project?
Yeah, so so my background is actually in maize genomics, so I come from sort of an agriculture background. So a lot of these models are very heavily informed by what's being done in both maize, but also in animal genomics is actually a closer system to trees. In terms of what it can bring into the other life science fields, obviously tree rings are, sort of a unique aspect of being a tree. But what I think you can really take into, especially the human side of things, which is sort of an equally intractable system in a lot of ways. I mean, it's easier to sample trees and we don't sort of have sensitivity issues. No one cares, which is nice. But on the other side, it also is sort of unique individuals in unique environments. And, outside of twin studies, there's not a whole lot you can do about it. And I think what we can bring forward is this idea of longitudinal measurements. Because what we have in trees that basically gives us all this power is that we can observe it, over time, and it's not unconfounded. I mean, these are, not independent observations that we have, yet the models still seem to work. So, I mean, if I were working in humans, what I would be trying to do at this point is trying to measure more longitudinal studies and working with that kind of data.
Could probably learn a lot from you there. Do you have other questions in the audience? Yes, John has a question.
That was amazing. I learned so much. So it may be too early to have this level of insight, but I'm wondering, how much have you already been able to understand molecularly about the adaptation? And so, I mean, you can kind of predict now for individuals where would they do better than they might do where they are. But could you extend this to, what would we need to do in order to design a tree for what the climate will look like 50 years down the road? And is there enough molecular correlation to say something about this, or is it too early? Or can you say something where the field stands at that level?
Right, I mean, so that's the flip side of it, right? So the prediction modeling is good for sort of breeding, and I actually really think, this is good for starting a breeding program. So if you don't have a breeding, obviously Sweden has a breeding program, so I'm not, in terms of that, it's less useful. But like, say you're in Central Europe and there are no breeding programs more or less, then you could basically get a jump start of one generation through this. On the other side, though, is the other side of association genomics is the actual, sort of GWA type modeling. And so this is, I think, getting more to what you're talking about in terms of understanding the trait architecture underlying all these adaptive traits. And so, yeah, I would say we're not quite there in terms of actually learning anything about biology yet. So we know it's working. We've sort of shown that we can find things, but we haven't dug deeply enough into it to even sort of start to identify differences in trait architecture in different sort of populations, which is something I'm interested in. So say you are having a breeding program, should we draw allelic variation from here, or is it really all shared? Or we just don't know any of this yet. But that's what we're moving towards really, really soon, actually.
Yeah. But maybe to follow up, is that a question of just working more on it and the data in principle is sufficient, or do you simply don't have the data right now to go deep enough on what are the traits of the architecture? But it's more actually mining it, basically.
Yeah, so I would say we do have the data. I'm not sure we have the population sizes yet. So that's what we're working towards. And actually, to be honest, we can sample way faster than we can process it. So I have a postdoc right now working on trying to automate this cross-dating. So you really need to make sure that you know which year is properly assigned to which ring, because otherwise your whole series is off if you miss one. And sometimes trees just don't grow for a variety of reasons. So this is sort of a really important process that the current gold standard is somebody who knows about these things looks at it and makes a decision, which is not very satisfying. So I have a postdoc right now working on trying to automate this, trying to make, essentially, as an analogy, some kind of blast algorithm for cross-dating that we can really scale this up. But yeah, I have a whole army of people working on cross-dating right now, but really we're trying to get these samples together.
Right. Yeah, and such automatic tools would be great for the whole forest community, right? So this could be
exactly.
impactful to develop.
Thank you so much.
Thank you.
So For our final talk before the coffee break, we will get to hear some unpublished results. So therefore, we will say bye to those who are on YouTube and welcome you back at 3 o'clock. But the rest of us, we will get to hear from Andrea Fossi.
Correct.
About Welcome back. I hope you enjoyed the coffee, and I heard there was a lot of networking at the posters and so on, so that's fantastic. And now we have the great pleasure to invite for our very newest, fantastic recruited fellows. So we have Mohammad Arif, we have Mariana Pires Braga, we have Chimney Twibedi, and Golnaz Taheri. Welcome. And 30 seconds each is what you will get. So we start with you, Arif.
Yes. Thank you so much for the time. So, yes. Oh, yes, 30 seconds. Yes, yes. So my name is Mohammad Arif, and my research is mainly focusing on the use of systems biology, data analysis to understand the progression of diseases. So as Ulla mentioned as well, that sometimes the time aspect is getting neglected in our approach. So that's what I want to understand. And we are focusing mainly on cardiovascular, cardiometabolic diseases and aging, and we want to use different omics data to understand their progressions and hopefully to find early treatment or new early biomarkers to get it stopped to progress. Thank you.
Thank you. Mariana?
Hi, I'm Mariana. I study butterflies, and they're sort of my model system for studying evolution of host-parasite interactions. And that works because butterfly caterpillars are parasites on their host plants. And they are a great system because comparatively we have a lot of data. So, for example, we know the phylogenetic tree pretty well for butterflies. So we know like which species is more closely related to which species. And we also have a decent amount of data for who are the host plants for each butterfly species. But a big problem is that our data is very biased towards Europe and North America. So where most butterflies are, we actually lack a lot of data. So in my lab, we combine development of methods for predicting interactions, and then we'll also go to Brazil, to the Atlantic forest, to actually test if our methods work and if we can then produce more data faster so that we can understand diversity in the whole world and not just in the temperate regions.
Thank you.
Hi, my name is Chinmay Dwivedi. I'm based at Umeå University. We are basically working on the human gut microbiota, and we're studying how microbes adapt to host health, primarily. But from a DDLS perspective, we are developing a new framework where we not only study bacteria, but all domains of life together and how they interact with each other, micro-microbe interactions, and also with the host. We're also going beyond species. We're trying to study strain-level associations and how strains interact with each other. One of the biggest problems in gut metagenomics is to connect taxa to function, because a lot of bacteria, they share function, right? So we're building a framework where we build bottoms up from genes to genomes, so we can actually study functional variation in the microbiota and how that links with health and disease.
Thank you.
Hello, everyone. My name is Golnaz, and I'm a computer scientist. I'm in the Department of Computer Science and Electrical Engineering at KTH. My team working on two different topics. The first one is to finding the mechanistic behind some rare or less studied hormone-driven cancer, and to find out why some of the patients shows resistance to therapy or showing some kind of secondary cancer development or metastatic behavior. And the second research topic that we are working on, that is drug, some kind of interaction prediction, but we are trying to be novel, and we are trying to incorporate more modality of data. So we are working with multimodal data, and we are trying to develop method instead of just be a user of some kind of tools and method. Thank you.
Thank you.
So thank you very much. It's nice to have you here. So we have prepared a few questions that we would like to, well, we are curious to see what you have to say about them. And not everyone will get every single question because we will not have time for that. But I will start with a question regarding basically your identity, or DDLS identity. So we aim to place DDLS in the very intersection between life science and data science. But how do you see yourself? Are you a life scientist who like to use data, or are you a data scientist who study biology? So if you start, Arif.
Yes, thank you for the question. So my background was actually from the computer science part of things. So I was working as a hardware engineer, like a software engineer for networking devices and stuff, but now I'm trying to bring that perspective into life science. So it's been, since I did my PhD, I've been trying to use how information transmission, how we can mimic that theory into the life science aspect of, especially on the disease part. So yes, I was a computer scientist, but now I think I'm shifting towards this intersection, exactly what you mentioned. So that's how I try to place myself as the bridge between the computer science and also the life science part of the research.
Fantastic.
Yeah. Mariana?
So I'm a biologist. I guess an evolutionary ecologist is the best way to define me. But actually next year when we go to Brazil is the first time that I'm going to produce any data. And I've always been collaborating with computer scientists. So I guess I've always been like the person in the middle that can talk to the field biologists, like producing data, and then the people developing tools. And I think for me, the most fun thing is to develop models that can then answer my biological questions. So yeah, I'm somewhere in between, I guess.
And Golnas?
I'm for sure computer scientist, but I'm really interested in biology, especially in cancer. So yeah, I'm a computer scientist, but I'm really interested in these biological related problems. So I need to have knowledge about this kind of stuff, but yeah.
Thank you very much. And I just love the idea when you describe yourself as the bridge builders and communicators, because that's exactly what we need. Another way to do this is via collaboration. So I have a question for you, Golnas. What does an ideal collaboration look like for you? Yes.
Okay.
Sorry, yes.
Yeah, perfect. No, as a bioinformatician, we really depend on clinicians, for example, for clinical data sets. So there is this continuous discussion with a bunch of clinicians because we need the clinical data to work. One thing about a good collaboration is to have open communication and constant communication, and to set the expectations right from the very beginning. I think that's what is the key for a good collaboration for now for us. That's working really well with a number of clinicians across the country and outside Sweden.
Beautiful. So thank you, Arif. And now, Golnas, do you want to comment on this as well?
For me, also, the good collaboration is somehow having close connection with clinicians, because we need data and we need a biologically relevant question. So because of this, we need a close collaboration with clinicians. So fortunately, we have this collaborator to some extent, and yeah, this can give us some kind of communication to work with them and find a common ground and finding something interesting with the help of them.
Thank you. That makes a lot of sense. So now I'd like to also ask the same question to you, Arif.
Yes. Yeah, so I agree with both Chinmay and Golnas as well, that especially myself, I'm not a trained biologist or a clinician, so I need that help from the experimental biologists or the clinicians. One, it's of course to produce the data, but also secondly is to guide us so that we are not only sticking to the mathematical side of things, but also on, okay, what is the real questions that you need to answer from this data, and then they can guide us through that. So it's not just merely we're giving them, okay, based on the mathematics, this is how it works, but then in the clinics or in the real life, it's not translatable. So that's why we need that kind of close collaborations with different aspects of the biology and clinical settings.
Thank you.
A follow-up question there. Could you elaborate a little bit regarding establish those collaborations? Is this something you have had time to do already, or something you're working on? Or do you see any kind of main challenges there?
Yeah, that's a good question. Actually, I'm pretty lucky that I'm with my current position now at the Wallenberry Lab in University of Gothenburg, that there are a lot of very good experimental, preclinical and clinical people in there, and I kind of established my niche in there. So it's kind of easier for me to talk. Okay, I have this unique expertise that maybe you are interested in collaborating. So it's been a fun journey too, and I think it's one of the most fun part that I get from this 1 year of establishing the lab. It's basically talking to people and trying to get them understand that I can give this value-added points to your research.
Thank you. So I have a question for you, Mariana. You work with a quite special setup and organisms, butterflies, and what do you see as the single biggest bottleneck for your specific field? Because you're talking about data generation as well. Is the lack of data, or is the lack of methods to process the data, or?
Yeah, so I'd say it depends. All of the above. So during my PhD, what I did was exploring different methods. So basically, we had these hypotheses that we wanted to test, and there was simply no good method for properly testing them. So I was just like trying different things until we developed our own models. And now, I collaborate with people at the Naturhistoriska Riksmuseet and at KTH. So hopefully next year, then things will like already be usable by people, not just by us developers. But then at the same time, there's also a lot of lack of data for some regions of the world, like I was saying before. So that's also important, and that's what we are also trying to do, both in the method side, trying to have pipelines that will speed up our data collection, but then also, of course, we have to go to the field and do that. And then also so I really like to talk to different people, and people have been gathering data in different ways. Yesterday, for example, we were at the Entomologiska föreningen for Uppland. So it's like amateur entomologists. And then I found out that, well, there's this huge butterfly collection in Gävle. Who wouldn't know? And but then putting all these different data together is such a huge challenge to then be usable for analysis. So I think that's one of the next challenges that I'm envisioning that's going to be coming my way. It's trying to use different sorts of data and combining them.
And do you see any way that DDLS can help you in that task? Maybe help you in addition to what we already are doing? Is there any particular wishes you have to help you to overcome these kind of hurdles?
Yeah, data center. Yeah. So. Yeah, all the from data storage to to making the data usable, making the data available for the community, all of that. I'm really hoping that we'll get to collaborate a lot with the data center to make that possible.
Great, thanks.
Thank you. I have a similar question.
Yes, you can go. You can continue.
Thank you. For you, Golnaz, actually. So now we asked Marina about her challenges. But if you were given unrestricted access to data tomorrow, what would be like the single moonshoot experiment that you would launch?
I really want to incorporate all the modality of data, all the information that I have, to find out what is the underlying mechanism for these, for example, cancer, and what makes them different. So if I have enough data, from, for example, different stage of cancer, I can design a method for it to find out what makes them different to different group of patients, to show different response to, for example, different medication, or yeah, this will be my moonshot project.
Thank you. So I have the same question for you, Arif. Unrestricted data.
That's a tough question. Because for us, mainly that the more data, the merrier. So then it's always good to have as much data as possible. And also what we are trying to do now, but it would be nice to have more data, is basically that we want to create digital organs, so that instead of having needing to use the real biopsy results or even model it with animals, so then we can create a very nice model that can represent the organs and how they behave in different different conditions, etc. So then that would help us a lot in shortening the research time span, etc. And also to save money as well, and less animal usage as well in the research.
So you're going to build the alpha organ.
Yes, a digital human being.
Fantastic. Do you have? We can follow up with the same question to you, Chinmay. What would you do?
So I would like to characterize all bacteria in the human gut. Right now, we assume that we have reference genomes for all species, and we believe that, or we assume that all diversity within a particular species is similar to the reference or species that we have. And that's not true. Different strains of a species may have very different functions. It's just that we don't know. Isolating them is a problem. Culturing them is a problem. Characterization is a greater problem. If I had all the money in the world, I would like to isolate, culture, characterize each one of them, and then probably conduct different trials to study their mechanistic role in the gut microbiota. Because right now it's mostly associative.
But don't they collaborate also? Or interact?
They interact also, and it will be very interesting to see how do they interact or compete. Yeah, it can go on.
Ska vi köra någon mer?
We're so efficient at answering questions.
You're so efficient. So we could. So now we were talking about unlimited access to data, but if we go back sort of to reality, what are your current bottlenecks? Is it access to data, or do you have other bottlenecks? Anyone wants to?
For me, it's accessing to data in a way that makes sense, and we can build upon those kind of modality to come up with some result. So for me, is having access to proper data.
So you would be looking then for collaborators that
exactly, because
could
of
provide
this,
you with
I'm looking
that.
for different collaborators who have these kind of data too.
Mm-hmm. I will remember that.
I think the biggest problem for us in our field is reproducibility of the results. We don't have common methods, and we don't have a consensus in the community to use specific methods, so everyone uses their favorite one. And the results may vary depending on which method you're using. That is one of the key reasons that we're going to develop a new framework where we want to integrate the pros of different methods while getting rid of cons of the rest. Yeah, I think data reproducibility is the biggest problem in my field, at least.
For me, I think it's both. I mean, I think we're closer, in the method side, I think we're closer to getting efficient inference methods for our complex models. But then, I guess, I would want more people to rear caterpillars on different host plants. And because the thing that we're really interested in, it's not just the realized interactions in nature, so the ones that you can actually observe. And we know that we don't observe a lot of the interactions that are happening, but also the potential interactions. They're not happening now, but they could happen, and they might happen with changing conditions. So basically, we're trying to predict which are the possible interactions for the future. And then for doing that, then you have to experimentally test those interactions and see if they work. And this is one thing that we're going to do, but we're only 6 people with limited time. So yeah, that type of data, it would be amazing to have more of.
Yeah, I guess I agree with everyone here. So the methodology limitation sometimes the issue, and also since we are looking at the specific like aging process, cardiometabolic diseases that are so variable from one person to another, that's also another issue to get a robust method to represent the variability in the human population. So that is something that we are working towards on. But yeah, but that's currently a big issue, because one variable, you change one variable, and then there's a ripple effect that there are so many things that is going on. But that's always the issue with the data-driven research, I guess. That we have to kind of oversimplify many different factors.
Thank you.
Good. I think time is actually up.
Okay. Then thank you so very, very much. And we would also like to wish you the best of luck, and I hope you will enjoy your coming years here within the DDLS program and really be successful in all your endeavors and moonshoots. So thank you very much.
Thank you.
Yes.
I have a clock that I can see there as well.
Okay, we are ready for the next scientific talk. And as Jan mentioned during his talk, the collaboration between DDLS and WASP. WASP is the another Wallenberg program focused on AI, is an important part. And we have a person here who will provide an example of such a collaboration. We have Joachim Jaldén here. Joachim is a professor at the Royal Institute of Technology, and you have been actively collaborated with the DDLS researcher in a jointly funded project.
That's right.
And the title of your talk is Restless Bandits and Self-Driving Microscopes. Very nice to have you here, please.
All right, thank you. So put myself on the spectrum. I'm an electrical engineer, actually. I call myself a computer scientist or an applied mathematician sometime. And I'm also from Gävle, although I didn't know we had butterflies of that magnitude. I chose a bit of a coy or mysterious title. I'd like to demystify it before we go any further. So I hope that the notion of a self-driving or a smart microscope is known to many of you. And if it's not, I want you to put yourself in the mindset of someone growing cells. The cells do something interesting some part of the time and somewhere across your experiment. And you want to take pictures of that interesting event happening. As many pictures as possible of interesting events, and as few pictures as possible of uninteresting events. And ideally, you'd like to have an AI that make those decisions for you so that you don't have to do them yourself, so you can scale up your experiment. So that's about the smart microscope. So what about the bandit? And the bandit is a nod to the notion of a multi-armed bandit, which is a term from reinforcement learning. And it builds on the name, the one-armed bandit, which is another term for a slot machine, where you pull the lever and the arm, and you get a reward from that machine. The multi-armed bandit is a thought experiment where you have a row of these machines. They all have different payout probabilities. You obviously want to play the machine that gives you the highest reward, but a priori you don't know which one it is, so you need to play all of them, and then you need to slowly transition over to the machine that will give you the highest reward. And that's an archetypical problem in reinforcement learning that has been studied a lot. So what makes the bandits restless is if that reward probability, so in the original setup, typically the rewards are fixed, you learn them in one shot, and that's the end of the experiment. Here, the reward probability changes over time. So if you haven't played the machine for a very long time, you forget about, or your knowledge about that machine becomes stale. So there may be an idea to play it again. And before going into our solution, I wanted to give some thoughts about modeling, and modeling in my context means mathematical modeling. So someone knows what this is depicting, shout it out now. A cow on the moon. It's a spherical cow, and it relates to a joke in the theoretical physics community that a theoretical physicist can model a cow. He just needs to assume that it's spherical first. And it's sort of a play on this notion that we tend to oversimplify things when we do modeling. But there's also a famous quote by George Box, who's a British statistician, that says that all models are wrong, but some are useful. So I'll present to you a ridiculously simple model for this setup, and then I'll try to close up by convincing you that it's actually useful. So mathematical modeling. We want to model this case of the smart microscope taking in images. So what's important in that context? So to me, what's important, it's space and time. So interesting things happen across space, so across different fields of use, but also within the field of view, and things evolve over time. So if you want to model space, something that you could do is you could divide each field of view into a grid, essentially, so a couple of boxes. So that gives you a notion of space, so things can happen in a location. And then you could say that a location can be interesting with some probability. Let's call it alpha for now. And if it's not interesting, it's uninteresting. And if you don't like the notion of a discrete space, you can also make this into a continuous space model. And the way that you would do that is you would let each one of the grids become infinitesimally small in a controlled way. And that would arrive at something called a Poisson point process. So that's a very common statistical process for modeling interesting things happening across space. Essentially, it just tells you that if you have a certain area and a certain intensity, you have the number of interesting events in that area is going to be Poisson distributed proportionally to the area and the intensity. And so the normal or the classical Poisson point process doesn't really convey the notion of a time, and that's important for this experiment. So what we do in order to capture time is we pick the simplest possible time-dependent model, and that would be a Markov process. So instead of saying that an area is either interesting or not interesting, we say that at each area or each box, we have a Markov process which can move from an uninteresting state into an interesting state, and then back again into an uninteresting state. We say that the probability of moving from the inactive or the uninteresting state to the active one is alpha, and the probability of moving back is beta. And you can play this continuity game here as well, both in terms of space and time. So if you let the number of boxes go to infinity, and you let the physical time transitioning or passing between state transitions become infinitesimally small, you get a model which is both continuous in space and time. But it's essentially a collection of Markov models moving in and out of in an interesting state. Right, so capturing the modeling, so this is a very simple model, obviously, but the niceness of having a very simple model is that it becomes mathematically tractable, so you can do calculations based on this model. So just to give you an example, I will not walk you through this equation, I will walk you through an equation a little bit later. So essentially, what this equation is just saying that if you've observed J interesting events at some time t, what is then the expected number of interesting events you should expect to see at some time t plus tau into the future, or some time tau into the future? And that's given by a relatively simple expression. But the main point is that we can characterize the statistics, and being able to characterize the statistics, we can also properly define notions of optimality. So we can phrase this problem in a way that we can rigorously analyze. So let's tie that back then into the multi-armed bandit problem that we have. So what do we have? We have arms, and in our case, the arms are tentative fields of use, places we could image in the experiment. We have a reward. We need to decide on what we try to maximize. And in our case, we want to maximize the number of observed Markov chains in the general case, that is in the interesting states. Or if we pose it in the life science sense, we want to see as many of the interesting cell events as possible across all fields of view. So what do we need to do in order to solve, what does it mean to solve the problem? So in a multi-armed bandit problem, a solution is an action policy. So a rule which tells us what action to take in the next step. That rule should ideally take into account everything that we know about the experiments, so everything that we've seen so far across all fields of views that we have decided to image. It should also maximize the expected reward in some sense. And it should be realizable. We need to be able to implement it, right? If we just pose the problem, that's not enough. We need to come up with a rule that we can actually compute. And the nice thing about coming with a simple mathematical model and making it in this formalistic setting is that there is a wealth of literature on how to solve this problem. So we don't need to come up with our new ideas on how the problem should be solved. If we can do the calculations, we also will have the solution. So one of the things which is very important with restless multi-armed bandit problems is something called the Whittle index. And I will walk you through this because it's an interesting concept and it also makes intuitive sense. So what is the Whittle index to begin with? Well, it's a real number that you can compute for each possible field of view. And essentially this number, it tells you how desirable is that field of view to image. Right? So K here index the field of view, it gives you a number on essentially how much should you want to look at that position in the experiment. It contains a number of quantities that are of interest. So this alpha bar over beta bar, so that's the probability of moving into an interesting state over the probability of moving out of an interesting state. And that ratio, alpha over beta, is essentially the expected number of interesting events that you should expect to see if you haven't done any observations yet. So the average rate of interesting events. And it appears in two places, in the first place and the second place. In the second place, it appears together with the number of interesting events that you saw last time that you looked at a particular field of view. And you see that it's minus the expected number. So essentially the thing in the parenthesis tells you, when you looked at a field of view last time, did you see more interesting events than you were expecting or less? And that's of course important. And then there is a weight factor in front of that. And what does this weight factor depend on? It depends on tau. Tau is the time that has passed since you last observed that field of view. And then we need to also factor in the dynamics. Is our system moving slow or is it moving fast? So you multiply that with a beta, so essentially the probability of moving out of an interesting event. So if you've seen an interesting event, how long can you wait until it will become uninteresting? And that enters into the negative exponent that you see there. So essentially when tau goes to infinity, when it has passed a long time since you last watched the field of view, you don't know too much about it, then this other term will go to zero and you just have the expectation, what you know statistically about that field of view. Then there is a tuning parameter that is particular to the Whittle index. So you can essentially decide about your reward. When do you want to have a high reward, now or later? And this factor gamma sets that. So if gamma is 0, you just care about the now. If gamma is closer to 1, you also care about the future. So sometimes you should image not to see interesting cells, but just to figure out that something interesting will happen in the future, right? So you can, by imaging, you can set yourself up for success in the future, and this gamma captures that effect. And then the policy is very simple. You compute this number, which is not very hard to compute, for every possible field of view, and then you pick the highest number, and you select to image that field of view. And then, of course, the relevant question is, does it work? It's a very simplistic model. Another way of phrasing it, how spherical are really the cows in this case? And of course, you can assess that experimentally. So here we have an experiment, and don't ask me about the technical details. There are people in this room that can tell you about that. But it's growing cells. They are stained with a cell cycle indicator, so you can directly observe from the fluorescent channel which state the cell is in. There is a bright field imaging. There is a green fluorescence for a couple of the states, and there is a red fluorescence for the G1 phase. So we set up an experiment here, and we say that we want to see as many cells as possible that are in the G1 phase. Then, of course, with every experiment, there is some practicalities to match the mathematical modeling with reality. We image everything in this case, and that's just so that we can compare to different policies across the experiment. The idea, right, when you run this in reality would be that you let the AI decide what you image and what you don't image. We also work with a fixed grid, so we divide each field of view into a 40 by 40 grid, and essentially if there is sufficient red fluorescence shown in a particular grid, we say that it's interesting, otherwise not. So we simplify it a little bit. There are these hyperparameters, the alpha and beta, which is dependent on the field of view in the method. We need to somehow set those. So we estimate those during the first 3 hours of the experiment, and then we run it across 26 hours. So we have 29 hours worth of data. And then we can see how it works. And it works well. You don't have to worry too much about the scale on the axis. You have to know that it's time on the x-axis and what's called cumulative regret on the y-axis. So when you do reinforcement learning, it's very typical to assess your methods in relation to an oracle method that gets to look at all fields of view, so get to play all of the arms and get to know the outcomes and can then post that observation, select the best one. So I mean, so that's an unimplementable strategy, but it gives you a bound on how well you can do. If you go with what would be the default method here, the round robin, so you image one field of view, you move to the next, the next, the next, regardless of what you see them, and you just collect all the data. This is how far you would be from the one that always imaged the one with the most interesting events. Second curve is one of the typical reinforcement learning algorithms, works very well for non-restless bandits. It doesn't work as well here. It's called Thomson sampling. It's a statistical method. I'm not going to go into detail on what it does. The next one, the yellow one, is called epsilon greedy. It essentially means that if you find a field of view that you like, you keep imaging it, but each time you image it, you flip a coin, and with probability of epsilon, you look somewhere else, just to make sure that something interesting is not going on elsewhere. And then you have these Whittle indexes, one for gamma equal to 0 and one for gamma equal to 99. And the absolute numbers, of course, doesn't matter too much. But what I want to show you with this is that there is a huge gain towards optimality by making the image in a clever way. So why does it work so well? It uses a probabilistic understanding of the problem at hand, and that is really helpful. And having this full statistical description, we can express what we know, but perhaps even more importantly, we can express what we don't know and what we need to know, right? And that informs the policy decision of what to image where. Obviously, no work is complete. Things can be done better. One of the things which is puzzling me and keeping me awake is that cells move. So clearly, the empirical experiment we've done shows that the method is applicable even if the cells move, and that's good. Otherwise, it wouldn't be very useful. But it would be very nice to model that effect as well. There are certain things, when you do the statistical modeling, you need to have descriptions of probability distributions. And if you do things wrong, those tend to explode in complexity. We haven't yet figured out how to account for cell migration in the model without making the model explode in complexity. So that's a very important open problem. Cells divide as well. They don't just go in and out of interesting states. When they've passed one round in the cell cycle, they typically divide, and there are two cells. That would be interesting to model as well. I think actually the migration is harder to model than the cell mitosis, but we haven't modeled either in this, but it still works. We have a very simplistic Markov model. It's just two states, uninteresting or interesting. one of the things we'd like to do, right, if you see a cell that is in a state that you can identify, maybe you will know that in some amount of time, it's going to be in an interesting state you really want to look at. And you can accomplish this by adding more states to the Markov model. So you can have, for instance, one state per state of the cell cycles, or you could also have several states for each state of the cell cycle, and that will allow you to. So essentially, if you have one state, the duration of that state always becomes exponential in these models. But if you have several ones, you can also get other distributions for the duration of a particular state. And that would be nice. There are these hyperparameters, the alpha and the beta. We view those as known in the model, and we estimate them from the data, but it would be very nice if you could learn those on the fly, because then they would turn everything into a completely hyperparameter-free method that you could just apply. That would be very nice. Obviously, you can do more extensive experiments. We only tried it in one case. If you want to show that it's generally very useful, you should try it across a much greater set of states, and I have to add this because my collaborators were a little bit disappointed that they weren't more neural networks. But you could put neural networks in there, right? So if you have neural networks that classify the cell state based on its morphology, and I know there are those of you in this room that have that because I saw that on the posters, that can of course be added. And one could also contemplate adding deep learning, for instance, data-driven ways of determining some of these hyperparameters. That could be nice. So at the end, acknowledgements. So to Jaume, that did all the mathematics, modeling, very nice. Wei, one of the DDLS fellows, he is the brain behind telling us that one should view this as a reinforcement learning problem. And also to Song Tao and Han Sao from Wei's group, who are the ones actually generating the data so that we could test this empirically in the end. That's that.
So thank you, Joachim, for a very nice example of how to use statistical modeling. Do we have any questions from the audience? Yes, Sven.
Thanks. Second time in a week I see this
presentation. Yes, I'm sorry for that. It was difficult to make two talks for this time. Yeah.
I was wondering about the occurrences of interesting events, so to speak. In many applications, they're likely to be spatially dependent, that they appear in certain neighboring fields and similar. Can you make the...
So in cell-cell interactions, you mean, or in a general area?
Typically, if you might have multiple fields and some of them contain a tumor, and that's where you see interesting events.
All right, yeah.
Can you extend this to sort of modeling dependencies spatially?
I guess the honest answer is I don't know, but if I should try to answer with a I do know, I would say, so if you can estimate the parameters alpha and beta on the fly, I mean, hopefully the alpha would go up for the fields of view where you have a lot of these interesting events. But I'm not sure, I mean, maybe one could have a more direct model of this. I mean, my own realization doing this, I did a lot of cell tracking, and Carolina will know we worked on this together. So mapping out data after you do time-lapse microscopy in the past. And I thought about it very much as how do I fit my deterministic mapping of migrating cells into the reinforcement learning framework. I think I've switched and think that it's much more important to have this probabilistic model, which describes the probability distribution. Because then it fits better with the reinforcement learning framework. So maybe one should invent a new probabilistic model in your case that captures, in some sense, the thing you are interested in.
Yeah, this is more of an intuition, but in some cases it seems likely that picking the neighboring field to one with a high Whittle index would be a good choice. But maybe that's not a principled thing to do.
You mean, so you would image around the area where... Yes, I mean, so that seems an intuitive choice. It wouldn't really be captured by this model. So I call it a spatial model, but it's, I mean, the different Markov chains are really independent. I mean, if you could capture, if we could figure out how to capture cell migration, maybe we could capture spatial locality as well in some way. But I don't know how to do that at the moment.
We have time for one additional quick question. Yes.
It's very hard to see people on the screen.
Yeah, great talk. At some point, I suppose, interesting events become not so interesting. So if you've seen 1,000 of them, is there some way of incorporating the kind of novelty aspect?
Yes, so that is modeled. I mean, so the Markov chain that is associated to each particular spatial location can both move into an interesting state and also out of an interesting state. And typically what you will see with this Whittle index is that, maybe I should, oh no, I shouldn't move backwards. So in that equation, so if you are fixed on a particular field of view, you image that a lot across time, and you start seeing that the amount of interesting stuff happening there decreasing, then the equation would naturally tell you to switch to another one. So the second term would become negative and tell you that this is now a bad field of view, I should go somewhere else. And then you would ideally go towards the ones where you expect a lot of interesting events, but you would only have one look at there, and if that was not confirmed, that suspicion that there should be a lot of interesting stuff there, you move somewhere else. And then when you find some place where there's a lot of interesting stuff happening, you will stick to that for a certain amount of time until the rate of increase. So you have this second term with the number of events watched last time. So if you image it consecutively, you sort of update that term, so that term doesn't vanish then. So this tau doesn't go to zero. It only goes to zero when you're not, so the tau, the time since the last observation, only goes to infinity for the fields of view that you don't observe. The one you observe, you sort of fix it at zero.
So, but if I see the cell cycle events for the first time, that's really interesting. But if I see it for the 10th,
you mean if it becomes uninteresting once you've seen the interesting event, that's, I think I have to think about that.
Yes,
very good point.
So let's continue this discussion during the break. So thank you, Joachim.
Thank you.
So our next speaker is a DDLS fellow, Louisa Huget. You are also an assistant professor at Uppsala University, and you will talk about bridging the last mile, microbiome enhanced forecasting of pregnancy complications.
Yeah.
Please.
Okay, I realize I have a different title there. I've also been in a course from 9 to 3 today. I let the organizers know and they invited me. I'm like, I can't speak today because I'm in this course until 3. They're like, that's fine, we booked you 3:45. So if I'm confused out of breath, you know why. So in my lab, there are 3 things that we're interested in and trying to get an overview of, and that's the combination of precision medicine, modifiable risk factors, and women's health. And in that little intersection is the human microbiome. And I love the microbiome because it is modifiable. Like, all respect to you that work with the human genome, but you're stuck with the genes that you're born with. While the microbiome we can do something about. And that's also where my title comes from, this bridging the last mile. So my proposal to you today is that the human genome, it works pretty well. The human genome, transcriptome, proteome, whatever level you work with, it's gonna explain a lot of our phenotypes. Much like our mass transit infrastructure, trains and subways and so on, will get you quite close to where you wanna go. But if you can't fix that last 2-3K that people don't want to walk uphill in the rain, they're not gonna use your amazing infrastructure, or in our case, we're not gonna get the full prediction that we could do. So my proposal here is that the human microbiome is that last step where we go from pretty good to an excellent prediction system, prognostic system, whatever it is. So all the data I'm showing today comes from 2 cohorts of pregnant women. They have very similar setups. So one is the basic cohort that is based here in Uppsala and driven by Alkistis Kalkido. And the other is the Svemami cohort started by Nele Brusselaars and Lars Engstrand at Karolinska. And in both cohorts, we have longitudinal sampling. And we're working with pregnancy complications. So of course, we don't know when we're recruiting what is going to happen to this pregnancy. All we know is that these people are pregnant. And they answer extensive questionnaires about their health, their lifestyle, so on. They deliver microbiome samples in early pregnancy, in late pregnancy, and then postpartum, including a sample from their baby's diaper. So far, I'm only looking at what happens in pregnancy, so ignore the baby. Svemami recruited over entire Sweden, so initially 5,000, almost 5,500 people answered the questionnaire. About 1,500 of them gave up as soon as they realized they were supposed to be scooping poop. But those 4,000 that sent us their first sample, they're quite loyal, and most of them stayed throughout. We're mostly sampling from the large regions, the big cities. Interestingly, Jämtland in every cohort is overrepresented. I don't know what it is about Jämtland, they just love participating. Basic is only recruited in Uppsala. But I'll start a bit with the basic, and basic is very much focused on mental well-being. So it stands for biology, affect, stress, imaging, and cognition. And then while the study was ongoing, we came to them and said, hey, can you include microbiome, please? So this work was done in collaboration with Mary Kimmel, who is a double specialist psychiatry and obstetrics, so she was perfect for this. And when we started analyzing, there was nothing there. There's absolutely nothing connecting perinatal depression to the gut microbiome, which is maybe good, maybe one less thing to worry about. Then when my student Ben turned from species to functions, and specifically to neuroactive functions of the gut, there we started seeing clustering, not only clustering in different groups, but also they do correlate with different clusters of symptoms. So both the symptoms clusters in terms of stress symptoms, depression symptoms, and anhedonia symptoms, and they in turn correlate with different processes in the gut, which was cool, but it's really, really far away from giving us something we can work with. And one big problem we have is that this cohort is not huge. And on top of that, we have a lot of missing samples. And especially we have missing samples from the ones that do have symptoms, because if you are depressed and overwhelmed with a little baby, maybe you're not going around remembering that project you signed up for 5 months ago. So how can we deal with this data, make the most of this data? And being what it is. So for this, we could collaborate then with Prashant's group, also here in Uppsala, Prashant Singh, SciLife Fellow, and his postdoc, Andrei. And the first thing they did is, how can we impute this missing data? So of course, there's a lot of methods around for imputing missing data, but microbiome data is especially annoying. It is very sparse. Most things are not present. And if you try using more classical methods like SMOTE, it's completely out of sample. It doesn't work. So Andrei started first with this part, which is the top process you're seeing there, but then putting an extra layer on that, can we use this for classification? So taking into account these imputed samples, can we then separate the people that are going to suffer from perinatal depressive symptoms and not? And these are not amazing results, I know. But what we could do that couldn't be done before was that we're predicting this a lot earlier. So there is previous work that is predicting perinatal depression with 90% accuracy, but they're taking already into account everything that happened during delivery and the first weeks post-delivery. While here we're doing this while they're still 7 months pregnant. So are we missing a lot of people? Sure. If the reason you're depressed is because you now suffer from PTSD because you had a horrible delivery, this is not in your gut. But if we can pinpoint these women that are at very high risk and start working with them before everything goes bad, we're already advancing. So Andrei is still working on this. I think he's also applying to some positions. Hire him, please. So to stay a bit on the gut-brain axis, this was a short project driven entirely by rage, because I was pregnant during most of my postdoc. And luckily, my office was very close to the loo. I could type, puke, and continue typing. But this doesn't feel very sustainable or very fair. And then we decided to, well, do something about it. So if we know that a lot of gastrointestinal symptoms come from our gastrointestinal health, why not nausea and vomiting of pregnancy? And no one had looked at that, which was disappointing but not surprising. So first up on the, do I have a pointer? Yes. On this corner, I know this is very wide and it's not the kind of graphs you want to see, but it is how microbiome looks like, and that's why we're working on the last mile. But basically what we're seeing is that the more nausea you have, the fewer species these women had in their guts. There's no difference in the distribution, in the evenness. The distribution is similar, but they just have missing taxa. And the fewer taxa, the more nausea. And of course, this could be confounded by a lot of things. So here we adjusted for how they're eating, other depressive symptoms, stress symptoms, and still the puke score, this is a technical term, is the pregnancy unique quantification of emesis, puke. The puke score is significantly correlated to the overall structure of their gut microbiome. And then finally, this is maybe the plot that you're most used to seeing. There is a lot of species that separate the women with the least nauseous symptoms from those with the most symptoms of nausea and vomiting. Some are known to be pro-inflammatory, some are known to have these neuroactive effects. Others we're still trying to interpret, but maybe we're getting somewhere with helping people with the absolutely most common symptom of pregnancy. But finally getting to what I promised, yes, the outcomes of pregnancy. And the first outcomes I'm showing are related to the size of the baby. So both very big babies and very small babies are at risk for a lot of things. If they're very large, they can have problems in delivery, they can get asphyxia, they can break bones. They're also at risk for hypoglycemia because they kind of been growing in this very high sugar environment, and when they get out, they can't cope. So that's the problems with being large. While the very small babies are more fragile, they don't have, they're more susceptible to infectious diseases, mostly. So then my PhD student Anusha started looking first into the large babies. And again, what she found was a difference not in the species, but in the functions. So I'm going to try to explain this figure that she made, and she explains it better than me. But this is the gut. And here on your left side, we have what happens when nutrients are incoming into the gut. So breakdown processes. Starch degradation, proteolytic fermentation, so complex food stuff getting into small metabolites. And then here we have the central microbial metabolism, where the bacteria are just growing and dividing and doing what bacteria do. They can get into cross-feeding with other bacteria, and then they'll end up with some end products, where those that are most associated with the health and well-being of us as hosts are these short-chain fatty acids, and especially butyrate. So what she sees here is that these women, so the green ones are the normal-sized babies. The purple ones are the very large babies. When they're coming in, there are some differences, so maybe a bit more starch degradation, some differences in proteolytic fermentation, but nothing that sticks out. However, for the women that produce adequate for gestational age babies, all these nutrients are getting into the final metabolism. They're being converted into small molecules that are important for our physiology, while this is being completely lost for this very large baby. So either they're getting lost into cross-feeding, and basically they're just, they're not being processed all the way into something that is metabolically meaningful, so you just get excess nutrients. And we do know there's no difference in the food intake of these women, as far as we can tell. We don't have super detailed food data, but they're not eating more sugar, they're not eating more fat, they're not eating less fiber. They are eating equally well, but this food is not being processed in the same way inside of them, and this is leading to different outcomes to their offspring. Then we turn this upside down, and now we're looking at the very small babies, and here it's the opposite of what we've seen before. So we don't have a difference in the number of bacteria in their guts, but the moms that will have very small babies have a very skewed distribution, or more skewed than normal. So they have a few bacteria that are dominating their microbiome, and the rest is in quite low abundance. Again, these images don't look amazing, but what we did here, because we're epidemiologists, is that we came up with a better model. So the baby size shouldn't be predicted by the microbiome alone. That would be crazy. It's by the microbiome and the maternal age and the BMI of the mother before pregnancy, whether she has had children before, whether she's drinking alcohol short before or in early pregnancy, whether she has polycystic ovarian syndrome, whether she gained enough weight during her pregnancy, whether she has hypertension during pregnancy. When we do that, the microbiome is properly significant. And again, it's the evenness, and those logs-odd ratio are maybe not entirely correct, but they are definitely significant, and there are definitely quite large differences in the odds. So again, we're looking at this last mile. So we take these women that for all intents and purposes have the same health and the same risks, and then what is making the difference between crossing the line into having a pathologically small baby or not can be found in their gut, both in terms of diversity and in the actual presence and absence of species. Here we still haven't gotten as far as pinpointing which species these are, but this is work in progress. And then finally, the most exciting one, the microbiome in preterm birth. So preterm birth is worldwide, but also in every country, doesn't matter how rich or poor it is. Being born preterm is the highest risk for a baby, in mortality, morbidity, consequences, anything. So we start again from the SVEMAMI. This is a nested case control study. We have 2 controls for each case. We are only looking at spontaneous preterm babies. We exclude the babies that are born term, but early term, so we don't have any gray zones. They are either preterm or they are full term. We also exclude twins because, of course, they have different risk factors. And then we matched our cases and controls on age, BMI, whether they've had a baby before. And because the pandemic happened in the middle of our study, and we didn't know if this would affect either their microbiome or preterm rates. So they are also matched in whether they delivered before or after March 2020. So again, at first sight, these are small differences. So here we're looking at the vaginal diversity. I've been talking about fecal microbiome so far, but the vaginal microbiome is very important for specific obstetric outcomes. And we have higher diversity in the vagina of women that deliver preterm. The vagina is also the opposite of the feces. You want very low diversity in the vagina. So this is as expected, but no strong effect. And for feces, again, kind of as we expected. So here it's the opposite, so it's lower, but as expected. Where it got interested is when it actually got into prediction. So on your left-hand side, the blue line is if we only use the vaginal microbiome, and it's a coin shot. The green line is if we only use the fecal microbiome, and it's basically a coin shot. The red line is if we only use questionnaires and what the women could tell about themselves, and here we have a 0.74 area under the curve, so we're getting somewhere. If we add all the microbiome to that, doesn't make much of a difference. We're in the purple line, 0.77. What Nicole did then, so Nicole's a postdoc at KI, when there was a PhD student at the time, and now a postdoc. What they did then was that they filtered this data to include only those data that were in any way predictive. So it does not have to be a high predictive value, just any predictive value. And again, we're talking about a very, very sparse data. So when they did that, we already went up from 0.77 to 0.86 area under the curve. And by including those global diversity data, we came up to 0.89 on the test set. If we look at what values are driving the separation, on the left-hand side, it's everything that is individual level, and they're pretty much what you expect: previous pregnancy losses, previous fetal death, previous miscarriages, things that you would expect are correlated to preterm birth. On the middle row, you have the vaginal species, and here I ask you to mind the x-axis. So all these species are by themselves more predictive than any of those high important risk factors that we knew about. And again, here on your right, on the fecal species, same thing again. Each of them is almost twice as predictive as the vaginal samples. So even if at prima facie, when we just look at the microbiome, there was very little correlation, once you include all the host physiology, the microbiome is making the difference into really pushing this prediction. Are you staring at me because I'm over time?
Yes.
Then I'm finishing here, because this is a good place to finish.
Thank you very much for a nice talk. We have time for a super quick question, if there is one. Otherwise, we will move on. I can't see if there are any questions. Okay, Sven, super quick.
So thanks, wonderful talk.
I can see it.
So hello, hi. Yeah, so to what degree can you control your gut microbiome?
Sorry, can you repeat?
To what degree can you choose what microbiome you have? Can you sort of ingest something or similar?
Yeah, so I mean, this is still very much an open research area. So we know when people move to a different country, it takes them about 7 years to be more like their receiving country than their country of origin. And we also know that most probiotic interventions, you have to be taking them continuously because they get washed out. But you would need to have a more combined intervention. So simply moving to a different country and not actively changing your microbiome or not actively changing your diet would probably not do much. But this is also very much about being able to pinpoint which species are of interest and really tailoring. So it's not any fiber, right? Right now you're saying a high fiber diet, but which fiber? Is it algae fiber or is it? So I think there's a lot of space for precision microbiome nutrition once we know what we're aiming for.
Great. Thank you very much. So we have reached the final point of this session, and this is, again, 4 short talks. This will be about the RDCPs, the Research Data Community Project. And again, as we heard earlier today, these are projects that aim to strengthen research around specific questions regarding data integration and data analysis. And as before, because the talks are short, the speakers will introduce themselves to save time. First up, we have Huang Sang.
Thank you. Hello, everyone. My name is Han Zhao. I'm a postdoc in Wei Ouyang's group. And today I'm going to present this proposal about AI's power smart microscopy and research data community on behalf of Wei, since he cannot be here today. So, AI power smart microscopy, it has great potential to transform our way to study cellular processes. However, we're kind of facing a fundamental challenge now. Most foundational AI models are extremely data hungry. To create a robust AI model, we need extensive live cell data sets. However, most of the open repositories now, there are lack of AI-ready data, which means AI models, they cannot directly learn from them. And another critical gap here is that we have very limited community coordination to systematically generate this kind of data. And so this is the gap we're facing. And this is also exactly what our research data community is addressing. Okay, so our vision here is to build a thriving research data community on making live cell data fair, to make the data foundable, accessible, interoperable, and reusable. And this is also to demonstrate the power of foundational AI models. To do this, we have 4 interconnected steps. Well, first, we're going to aggregate community data. So we're collecting data and put them into a united repository. And second, we're making these data AI ready. This will mean much more than just storage of data. For example, currently, we're adopting OME-ZAR to allow for sufficient storage and streaming of data. We're also curating metadata, and we're also creating some comprehensive documentation, such as README file, which allows both humans and AI to work with. And the goal here is seamless data access. And the third step is to train a foundational model thanks to the rich and diverse data set contributed by the community together. And the last step is to enable smart microscopy. So where AI can control the microscopes in real time and to adjust based on what it sees. And this will open some new experimental opportunities in microscopy. And the key messages here is that we're tackling this data challenge as a community together instead of as isolated labs. So here is, I'm going to show some concrete outcomes we can deliver. Unfortunately, I cannot click here. We do make a demo site. So the first delivery is the FAIR Data Hub. It's a national data repository with integrated compute access. This means, as a user, you can not only access the data, but you can also run some analysis and AI inference directly on the platform. And the second delivery is the foundational models, specifically for live cell imaging. And apart from the model, we are also going to make public the model weights and the fine-tuning workflow, so everyone benefits from everyone's data. And the last delivery here is the demonstration of the smart microscopy. Currently, we're building a system where AI can adapt or adjust the parameters during image acquisition based on biological events it can detect. And yeah, this is the demo site. If you're interested, you can open it. I can't click it now, unfortunately. So community engagement in this project, we are planning for 3 phases from 2025 to 27. In 2025, which is now, is the launch and the connect part. It's the kickoff of the project. We are planning and recruiting a data curator, and we'll also connect our facilities and set up all the infrastructures for this one. And in 2026, we build and engage. We're deploying FAIR Data Hub platform, and we will also collect and start the curation of these data sets. More importantly, we're going to co-organize some community events, such as some conferences or some symposium. The goal is to expand the community and demonstrate values. And by 2027, we scale and demonstrate. So we host some workshops, some hackathons, and some challenges, like model training challenges. And we're also going to showcase smart microscopy demos. And by 2027, we're also going to release the data sets and the models. So by the community engagement, each phase will progressively expand the community from coordination to contribution, and finally, we co-create. This is the expected impact we're at least expecting. So of course, the first, we're building a national data infrastructure that connects the facilities, the researchers, and also AI developers. This is also a network with at least 10+ partnerships, expanding from universities to imaging facilities. We're also positioning ourselves in a European leader in AI microscopy. By delivering the first live cell foundational AI model, we are establishing ourselves to the forefront of AI bioimaging here. And for broader impact, we're hoping to help with broader and faster biological discovery in other research fields, such as cancer research and drug development. We're hoping to extract deeper insights from microscopy experiments by making data fair and making models accessible. Yeah, so that will be all from my proposal. Thank you for listening.
Thank you very much. So the next speaker is Carolina Välby.
Thank you for letting me back on stage. Should I click forward to get to my slides, or how does it work? Yes. Yes. So I'm going to talk about the Tissue Maps Research Data Community Project. So I'm going to start with some data. So this is a piece of a breast tumor, and what you can see on top here is spatial transcriptomics data showing estrogen receptor. We can also show cell type deconvolution, and here we see the lump C cells, and you see that they sort of correlate with where we have changes in the morphology. But what's maybe even more interesting here is that on top of this, we can also put layers with mass spectrometry data. So this is the metabolites in the tissue. So now it's not very exciting, we just see this gray part here, but we have all these different peaks in the mass spec data. And I happen to know that there's one peak, a tiny little peak there that's quite interesting. So I can select that one and then go back and show that particular peak on top of my data. And also then I need to hide the other data. I got this data yesterday, by the way, so that's why it's a little bit new to me as well. But what you can see here now, this is ascorbic acid in the tissue. And it actually then correlates where we have these changes in the tumor morphology. So this is what we want to do. We want to maximize the power of discovery from spatial omics data by providing a fast and flexible and interactive way of visualizing and annotating spatially resolved data, and also sharing data via this portal. So this is a web-based tool that we have developed for data sharing. And a big thanks to Marco for helping me yesterday to get this up and running. And also, of course, the BAF team. And I must mention that the SciLife Lab Serve and the data center have done a fantastic job in making it fast and efficient to spin these visualizations up. You can do it very quickly. So this is all types of spatial data that we want to work with. This is another example of spatial transcriptomics. And all this data consists of it can be layers of images, it can be markers, it can also be annotations. And the sort of visualization tool that we have built here is similar to Google Earth. So you can have data from many different patients and look at the data at the same time. But then if you have this much data and you want to apply your calculations or your models to it, you might not want to work with everything. You might not want to download terabytes of data. So we want to make it really efficient to cut out your piece of the cake, the part that you care about, so that you can do your thing there. And then what we also want to do in this project is to continue to develop this web-based tool, and also to align with new data formats in this field, and also simplify the contribution of new data from you all, and also make it simple to add plugins to this tool. These are some things that were recently added, this exploration of the mass spec data. We can also add information on differential gene expression and so on and so forth. We can also show measurements extracted from the data in a UMAP space. So we have the feature space on the one side and the data on the other side, and then we can interactively move between the two to figure out what the cluster and feature space would look like in the tissue space. We also work with proteomics data. These are cell classifications based on protein expression. And the two classification results that we can compare here. And whatever is on the diagonal is where the classification results agree. And then if we have off-diagonal classification results, we can go in and look at the raw data and try to figure out what we might need to adjust in our models. And finally, I just wanted to show this super simple data set. Oops. I wish I could play that movie, but I guess if someone back there can click on it, it will run. But in this case, we have different serial sections of the same biopsy that have been stained in different ways. Someone is trying to help me. Thank you. Yes. So it's the same biopsy. and 3 different slices that have been put on top of one another so that we can then compare very simple stainings, but just having them on top of one another and being able to flip back and forth and move around can tell us a lot about what kind of learning we might want to apply to these images. So that was my last slide. And I'm really hoping that this will bring something useful to many of you that are working with spatial data. Thank you.
So thank you very much. Next speaker, Sara Kalucha, please.
Thank you. Yeah. They were so pretty, I wanted to keep them. So my name is Sara Kalucha. I am willing to bet I'm the oldest sociologist in the room. But today I'm here as a registered researcher and a part of the Fosh Connolly team at Critical Microbiology in Umeå, of which I am a part nowadays. And to talk about critical microbes. So critical microbes, this project stems from an underlying issue that has come up over and over again today, which has been interesting, and that is the issue of fragmented data. So in this case, it's about the issue of fragmented infectious data. So the infectious data that we have today in Sweden is fragmented across all the 21 regions that we have. So all the different clinical microbiology labs have their own data, and if you want to access that data, you have to apply to each of the clinical microbiology labs and get access to that data. It's a whole process. If you've done it, you know it. And that is a big bottleneck for research. And what we want to do with critical microbes is to take away that big bottleneck and basically do a big chunk of this brunt work and create a national outcome database for infectious data to combine infectious data, bacteria, viruses, fungi, and parasites into one big database. Because this issue of fragmented data is really an issue both for research and for the regions, and this has been highlighted both by regions and both by individual researchers, but on a strategic level. So what we're aiming to do with critical microbes is to create whole population coverage, so all ages, everyone in Sweden, linking to other population registers. So not only this outcome data, because what are the outcome data if we don't know other things about the people? So we're linking it to other healthcare data, other healthcare registers, but also to demographic data. So we have, this is also building upon a different register database already in place. And we will have to work extensively with data harmonization. We have a data manager in place that will work with this. I will also work with this. And this will be to harmonize across all the labs, but also to create measures that can be used across studies to ensure comparability across studies. And this is then, in the end, for the end goal for this to be a database that can be accessed by those of you who might be interested, because we're not creating this for us. I mean, hopefully some of us might be using it, but the big end goal here is to create something with fair access for everyone who has ethical approval to access through secure measures through the Umeå University, so that we can utilize this, all the work that we're doing now together. Because in the end, we are really passionate about this, from data to better health, that we are really convinced that critical microbes fills a critical gap in our ability to study infections and their consequences, both long-term and acute consequences, and that we can increase our pandemic preparedness and also inform health policy and identify vulnerable groups in society using this data. But it has to be in this larger format to be able to do this, but we also have to do it together. So, yeah, that's what I had.
Thank you very much. We have a fourth and final speaker, Tobias Anderman, please.
Yes, thank you. And hello, everyone. I'm Tobias Anderman from Uppsala University, also DDLS fellow here. And this RDCP is about meta barcoding data. So we call ourselves the meta barcoding network, the Swedish meta barcoding network. And just to give a little introduction of what type of data we're talking about environmental samples. So environmental samples can be things like soil or water or air. And we, in many research groups in Sweden, we extract DNA from these environmental samples to sequence it and then map it back to reference databases to understand aspects about the local species community. Similar to how microbiome samples are being taken, what Louisa showed, we do this also in the environment to basically get an idea of the species communities at different sites. Now, there are some challenges with these data, which basically have to do with the data availability. So many of these research projects, they at best, at the end of it, put the raw data up on the European Nucleotide Archive. And the issue with that is that these data, even though technically available, they're not really fair. You can't really search there very well for occurrences of certain species. Often important metadata is not present. So we need to do something about making these data more available and comparable. And about the comparability issue, there's a lot of data generation of these environmental and meta barcoding data sets that is not standardized, sometimes with a good reason because they're done for different purposes. But there's a lot in the sampling protocols, in the lab workflow, and in the bioinformatic processing that could benefit from more standardization and at the least of better metadata to understand how these different samples were generated. And that's basically what we are tackling in this RDCP. So we are a group of more than 30 PIs across Sweden that are working with meta barcoding. And some of these PIs in this group have actually been essential parts of developing methodology, globally relevant methodology and databases for meta barcoding data. So in Sweden, we have a bit of a very active community, but there is not actually a community. So we have many different research groups that work on it, but we see the need to organize as a research community. And our idea is to provide a hub for eDNA and meta barcoding data expertise. This is both for researchers who want to start using this method, but also actually for stakeholders outside of academia, where there's a lot of interest in using these methods for example, environmental assessments. So different environmental agencies or public agencies are actually interested in learning this method, but at this point it's still largely contained in the academic silo. So we want to provide this hub, but also help define and share best practices and sampling protocols. This expertise exists, and we just need to consolidate it. And our idea is to have a web page where there is going to be some information about general considerations when working with these data, how to process, how to store them, but then also standardized or protocols that we will upload on protocols.io. Some of this already exists, but there's certainly more work that needs to be done there. So ideas generally make eDNA data fair and more comparable. And for the processing of these data and making them truly fair, we are envisioning this a scalable data stream using components that actually exist already and that have been produced here in Sweden. So we have this data processing pipeline called AmpliSec. which does all of the processing from raw data into lists of annotated sequences of OTUs, operational taxonomic units, or ASVs. And then we also have a spatial portal where we can actually upload this data and where you can run a blast search and see this sequence that I found in my backyard in Sweden has already been found in these places in the world. So we can actually start really seeing interesting patterns, spatial patterns about the biodiversity and the sequences we detect. At the moment, even though these tools exist, they are still very devoid of data. So we really need to fill these, this portal with more data. And then we have ongoing conversations with GBIF. That's the largest global biodiversity infrastructure. And here in Sweden, we have a head start at the moment, having these tools and this way of spatially displaying eDNA data. And there's a lot of international interest in developing scalable ways of doing this. So with such a data set of Sweden-wide biodiversity inventories, we can really get a good overview of, we can basically really map the biodiversity in all organism groups in Sweden. And this can help us building a good reference database where we can store operational taxonomic units or ASVs that we detected in Sweden. And we can actually either use that for taxonomic annotation, or in case of hidden diversity, species that have not been described yet, we can assign these sequences that we find, which we don't know exactly what species they are, but we can use a consistent ID to compare where in Sweden have they been found. We can start making some conclusions about their spatial distribution, maybe about their ecology. So this will really fill our gaps very quickly in biodiversity mapping in Sweden. So this is also excellent data for predictive models like the ones we're using in our research group, for example. And we can infer maps of species richness, of species distributions, and also do this for the dark diversity of undescribed species. So that's the big vision, and really all we need is to organize this community and get these data together. So I think there's a lot of value in that. So yeah, speak to me in the break if you're interested. Thank you.
Thank you very much. That was the last speaker of this session. So I think we should thank all those speakers this session again with an applause. It's now time for the next poster session. Those with an even number should present the posters. We will also be served some refreshments in forms of drinks and snacks to keep us going. Tomorrow we will start at 9 o'clock in the same room, so please be on time. And have a nice poster session, and see you tomorrow.
Speciellt om tänkte väl lämna oss.